Epigenetics in cancer, a hematological perspective. So what is epigenetics? And epigenetics is defined as heritable changes in gene expression that are not due to any alteration in the DNA sequence. Epigenetic modifications are placed by epigenetic writers and removed by erasers in dynamic but highly regulated manner. Over the past 2 decades, the connection between cancer and epigenetic regulation has been a promising venue for research. Many of these discoveries were initially described in context of hematological malignancies and acknowledging significant exceptions. Their counterparts in solid tumors have not been so easy to demonstrate. So, epigenetics and the hematological malignancies. So here we have epigenetic regulation, dysregulation, and therapeutic targets. With the availability of NSG, ChIP-seq, and CRISPR-Cas9 technologies, we now understand that the epigenetics involves a complex and dynamic interplay of writers, erasers, and readers, which act not only on promoters, but on many regulatory elements, including enhancers and repressors, forming a three-dimensional network of regulation. So this figure here is more of a visual to understand what they are talking about in the next upcoming slides. But just to break it down a little bit, we can see that the DNA here in red forms a complex with the histone proteins, which are light blue, to form these nucleosomes. And each nucleosome consists of DNA wrapped around a unit of 8 histone proteins. The epigenetic marks are placed both on the DNA and histones by epigenetic writers, which are in green. And you can see those green boxes. Epigenetic marks can be removed by epigenetic erasers, which are specified in the red boxes. Epigenetic marks are recognized by epigenetic regulators through a special reader domains, which are here in blue, which lead to the recruitment of epigenetic regulators to DNA and histones. The complex epigenetic regulatory program is disturbed by hematological malignancies by mutations in these epigenetic regulators, and those are shown by the red thunderbolts. In orange, we have therapeutic approaches that were approved by the FDA, and in yellow, we have therapeutic approaches that are still under investigation. So, role in cancer. Epigenetic dysregulation manifests in cancer with global DNA hypomethylation causing genomic instability as well as silencing of specific tumor suppressor genes and of microRNA genes by hypermethylation. Large-scale studies of DNA methylation have detected extensive hypomethylated genomic regions in gene-poor areas in cancer cells and demonstrated that the degree of hypomethylation of genomic DNA increases as the tumor progresses from a benign proliferation of cells to an invasive cancer. Global hypomethylation promotes tumorigenesis by the generation of chromosomal instability, reactivation of transposable elements, and the loss of imprinting. In most solid tumors, epigenetic mutations are rather rare and they are mainly found in hematologic malignancies, rare childhood cancers, and highly aggressive solid tumors like glioblastoma multiforme. Much of what we know about the epigenetic dysregulation in cancer has been elucidated by studying hematologic malignancies because most direct epigenetic mutations are found in hematological cancers. So here we have epigenetic dysregulation as a hallmark in cancer. So disruption in epigenetics has been found to collaborate with these hallmarks in cancer development in many ways. This figure shows an overlay of the proposed cancer hallmarks with the recurrent genetic mutations of AML with epigenetic dysregulation at the center of the complex interactions. So certain mutations seem to collaborate while others are mutually exclusive. For example, there is a strong association between mutations in the epigenetic regulator DNMT3A and FLT3, which is activating signaling, and NPM1, which is a tumor suppressor. On the other hand, mutually exclusive exists among transcription factor fusion genes NPM1, RUNX1, TP53, and CBPA. So the new research focuses on the intersection of cancer epigenetics and newly characterized cancer hallmarks of cancer immunology, metabolism, and alternative mRNA processing and splicing. So the immunosuppressive microenvironment and the epigenetic alterations are known to silence and downregulate all steps of antigen processing and presentation machinery in cancer cells, including the tumor-associated antigens, human leukocyte antigens, and accessory co-stimulatory molecules. Epigenetics has shown to upregulate all the elements in the antigen presenting machinery. For example, the expression of tumor-associated antigens, which is TAA, MHC1 and MHC2 molecules, as well as co-stimulatory surface markers like CD40, CD80, and ICAM1. There's significant evidence there based on preclinical in vitro and in vivo models supporting the combination therapy using epigenetic modulators and immunotherapy. So, combination strategies. In combining epigenetic agents with cytotoxic chemotherapy, the reactivation of tumor suppressor genes and restoration of DNA repair pathways by epigenetic drugs results in a more chemosensitive cells. These cells can then be targeted by another type of therapy. Initial studies combining epigenetic agents with chemotherapy showed disappointing results, though further studies should suggest that the timing of epigenetic therapy matters and that it might be able to reverse resistance to chemotherapy. So in conclusion, we've come a long way in understanding the epigenetic network from the initial model of epigenetic regulation. DNA gets methylated, recruits histone deacetylases, and the two-system button down chromatin and silence expression. The impact of epigenetic alterations in cancer is also complex. Many mutations in epigenetic writers, erasers, and readers have been identified as promoting cancer development. Furthermore, epigenetics has been recognized as lying at the heart of multiple hallmarks of cancer, interacting with cell cycle promotion, cancer metabolism, neoangiogenesis, and the immune system. As epigenetics is recognized as a very early driver for cancer progression, this holds promise for both improved early diagnosis and the therapy of cancer.